Hi everyone, my name is Danielle Chou and I work at a synthetic biology company called Zymergen, and we're in Emeryville, California. I'm a solutions engineer at Zymergen, and I'm going to be talking to you guys about how we use Jupyter notebooks at the intersection of robotics and industrial biology. Just to give you a brief outline of what I'm going to talk about today, I'm going to first give you an introduction of what Zymergen does and some basic biology terms. I know some of you guys probably have a biology background, but for those that don't, I'm going to give you an overview of some terms that I'll be using. I'm going to talk to you about what problems that we're trying to solve, and I'm going to go into a specific example of a problem that, as a solutions engineer, we had to solve. And finally, I'm going to be talking about how we use Jupyter notebooks to solve these issues. So you're probably wondering what a solutions engineer is. And so I know that term probably means a lot of different things at different companies. But at Zymergen, what it means is that we create rapid prototypes that we deliver to the scientists, and that eventually, if the tools are adopted, they'll get integrated into our production code. And so we kind of sit between the scientists, which are our bioengineers and our chemical engineers, and the software team. So we get to hear a lot of the scientists are essentially our customers, and so we get to interact with them on a day-to-day basis, which is really cool as opposed to other roles where your customer is out in space somewhere. And so we create these rapid prototypes, and we give them to our scientists, and the production code is what we call LIMS, and that stands for Laboratory Information Management Systems. So if you've worked in a lab before, you'll use a LIMS system probably, and this LIMS system is used to track our samples, to track experiments, etc. So to go over some basic biology terms, a strain is strains are groups of organisms with trivial differences in their genomes. So here is a photo I'm sure you guys have all seen, like some DNA. And if we zoom in on this DNA, you'll see that for the most part, this genome should be the same. It's all corn genome. But if you zoom into a specific part of that genome, you'll see that there could be these small genetic differences in the genome, and that results in these different looking corns. And so one small difference in the genome can create like a really different physical expression in corn. So our company is called Zymergen, and that comes from the term Zymology, which is the science of fermentation. And fermentation is used to create materials and molecules. And you're probably most familiar with fermentation in terms of making beer or cheese, but you can make a bunch of other materials through fermentation, like insulin, like food additives, and like fuel. So fermentation is really important to us at Zymergen. And specifically, what does Zymergen do? So we provide a platform with software and automation to rapidly improve strains through genetic engineering. So how it works is we'll have a customer come to us with their strain that produces some industrial fermentation product, and we make a bunch of new strains and a bunch of genetic changes, and we want to figure out how to improve their economics of their strain. So the way that fermentation works at these giant fermentation companies is they're in these massive tanks. And in order to test out on these massive tanks would be incredibly time-consuming, would be really costly, really resource-intensive. And so with our platform of attempting to make lots of strains and finding the best ones, we need to start at a smaller scale. And so we might start in something like a tube here, or like a 96-well plate here. And so these 96-well plates, if you've worked in biology, you'll know that 96-well plates are kind of where we do a bunch of our research and where we will test our strains. And so these might have like a milliliter of liquid in them, probably less, and they're what we use to do our testing. Another term you might hear from me is pipetting. So pipetting is through the use of a pipette. There is a vacuum that you can use to drop liquid and then release liquid. And so we use these pipettes to do biology research. And traditionally, they are done by hand. And if you're going really old school, they were done by mouth, we don't do that anymore. So yeah, it's scary. So there's a term that we use at Zymergen called hitpicking. You might also have heard of like rearraying or cherry picking, and So what happens is when we get, when the upstream teams who have made our new genetic strains bring us our samples, they bring them in, here's a 96-well plate, like the one you saw before, and we want to test these strains. So we want to do a bunch of testing on them, but in this plate, and these aren't actually strains, but you can see that the samples that are closest in color are in wells closest to each other. And if we're going to be doing a bunch of testing, we don't want our strains, our samples in similar areas on a plate. And so this process of picking samples from a source well to a destination well, and from a source plate to a destination plate, is what we call hit picking. So here is our destination plate, and you'd see that we might move our first sample to one part of the plate, and our next sample, which might be the most similar sample, to another part of our plate. So why do we hit pick? There is a surprising, despite the really controlled environments that we try and have in biology, there is still going to be a lot of variability within a plate, and that's because plates can be exposed to certain elements. There might be some evaporation going on in different parts of a plate. There might be temperature differences, and you can see, or from that last plate that I showed you guys, a lot of times people won't even put samples in your outside wells because typically those parts of the plate might have more exposure to elements, et cetera. So we want to move these samples around our plate to approve that the environmental conditions of the plate aren't what's affecting their performance, because we want to see how these strains are going to perform on our plate. And we want to be able to increase the chances that if we see a strain that's performing well, that it's performing well because it's a good strain, not because it happened to be in a part of a plate that had conditions that it liked to be in. So there is the idea of hit picking, and then we have our hit pick plans. And our hit pick plans define which source wells should go into which destination wells. On your right, you can see that that's a liquid handling machine. So the way that we do pipetting, or a lot of the pipetting that we do now, is done by a liquid handling robot. And this makes our lives way easier because pipetting, as you saw, is a very manual process. It's very error prone, and it's nice to have these machines that you can tell them, okay, move these liquids from this plate to this plate and do the work for me. And I was really excited when I saw these at Zymergen because my background is in biology, and when I did biology in undergrad, I had never seen one of these before. So these hit pick plans are pretty straightforward. At the bottom of this slide, you can see an example of, okay, here's my source plate ID, my source plate my source well, my destination plate ID, and my destination well, and then the amount of volume that I'm going to pipette. So this volume is really important because the robot and the pipette tips that you use determine how much volume you can draw up and release. And so we want to make sure to tell the robot this is how much volume you should use. There are a lot of challenges in creating these hit pick plans. So when I came to Zymergen, most of these hit pick plans were done manually, which is not very efficient and very time consuming. There was little standardization across teams. So within Zymergen, we have a bunch of different clients and projects, and they all might have different ways of creating these hit pick plans. And so they were all working on independent processes. And finally, there were some complicated Excel tools. And what I heard from one of the scientists was that, there's one person that created this Excel tool, and the rest of us run it, but we don't really know what it does, and we wouldn't be able to recreate it. And so that was a little scary. So this became a project of the solutions engineering team. And we had a few goals in mind when we set out to create this hit pick tool. We wanted to have a simple interface because a lot of the scientists aren't really used to seeing code. We do have a diverse range of we do have people that write a bunch of their own scripts, and then we have other people that don't really want to look at scripts. So we wanted to create a simple interface that they could use. We also wanted to have clearly defined error messages for them. So we wanted to be able to anticipate what sort of input errors the scientists might have, or make it easier so that they wouldn't have to look at a stack trace to understand what was going on. So we wanted clear error messages. And then finally, we wanted to use minimal engineering resources. So the solutions engineering team was made to be able to quickly prototype things. And that means we want to not have to use resources from outside our team as much as possible. And so that was really important when thinking about this tool. And that's where when we noticed that there was a lot of value add in using Jupyter as a solutions engineer. When I first joined, I wrote a command line tool, and getting the scientists to adopt the command line tool was much more difficult, and Jupyter provided this much more tangible, user-friendly interface. Jupyter makes it easy to standardize tools. So what's nice about using things like the widgets is that you can predefine what sort of input the user can put into the tool while still giving them options to enter. And the use of Jupyter empowered the solutions engineers to quickly prototype. So none of us have UI backgrounds, and it's nice to use Jupyter as our UI. And finally, if the tools were heavily adopted and the users liked them and the software engineers thought they were something that we could adopt into our LIMS system, then they would get translated to production code. Next, I'm going to go over some of the key Jupyter functionality that we used. And the overall goal, which I've kind of explained already, is that we wanted to create a user interface for the user. So we wanted to hide a lot of the code from them. So one, we used magic commands, and that helped with the simple interface. We used a lot of Ipywidgets, and I'll go through a few of those with you guys, and that helped with the user input fields and the clear error messages. And finally, we used the fact that if you enter stuff into a cell, you can actually see a visual of it, and that was really good for our users to get them to trust the tool quickly, because although the Excel tools were really complicated to use, they were also what they were used to, and so we needed to get them to trust our tool and adopt it quickly. So I think I've seen a good amount of magic commands in previous sessions, but just as a reminder, they're used similarly to commands in the command line. You can identify them through the percentage sign, and what we use them for is to hide a lot of our code. And so this top cell here is all that you see in our notebooks. Everything else is widgets that people put stuff into. And we use the custom extensions that we put in the IPython extensions folder. And if you name them load IPython extension, it's really cool. You can call them, and then they just appear. And so this is just to show you guys that one line within this function is all that you need in the IPython extension for us, and that just loads the hitpick interface. So next we used a variety of Ipywidgets. And so one widget that we really enjoy using is the tabs widget. And this makes a really user-friendly menu like interface where you can click through the tabs from left to right and input the necessary data. And so here you can see we defined our tab names, which will allow the users to do a variety of things. And then within that, the child tabs are actually widgets themselves, and those are all different types of widgets. So this is what it looks like to them. And so this isn't live, but you can go through and click on the different tabs and see what you need to enter. It's also really nice because we have, this first tab is just instructions on what the user needs to do. And then we also use the HTML widget to embed the URL, which links to our Confluence pages. So we really like, as I'm sure many of you do, using Confluence pages to do a bunch of our documentation. So if this documentation isn't enough, then you can go to the Confluence page and read in more detail what's going on. So here is one of our input tabs. So this is the replicate selection. And so when you're creating these hitpick plans, traditionally you don't just, you want to have several copies of your different samples within a plate. And so I won't go into the details of what these mean, but you you use these drop downs to define how many replicates of different sample types you have. And so this is really nice for us because we know the expected number of replicates or a reasonable number of replicates, and so we give them these options, which might be project dependent, but we restrict those options, and so they can enter, 0, 1, 2, 3, 4, something like that. The next tab I'm showing you is the volume tab. So the volume input, it defines the sample volume pulled from the source wells and dispensed into the destination wells. So when I showed you the liquid handling robot, we talked about how you drop liquid and you need to know how much liquid to drop. So this bounded float text widget is really nice for us because we know the specific parameters that the robot can pipette, and so we give all of those options to our users. So we know that at a minimum, we can pipette up 1 microliter, at a maximum, we can pipette up 100, and we can pipette to the 0.1 microliter. So we give them all those options. If you enter something that's over the max, it'll bring it down to the max. If you enter something under, it'll bring it up to the min. And so this is a really nice feature for helping create this Hitpick plan. So finally, at the bottom of the instructions tab, there is only one other icon, and that is the submit button. And we love the submit button. We use it for a lot of our solutions engineering projects. And how it works is that once all of the parameters are defined within the different tabs, the users can hit the Create Hitpick Plan button to submit the code. So here I've included, this is how you create this button. You can choose what kind of icon you want it to have. You can add a description, and then upon clicking the button, you can call the function that you want to use. And so we really like this feature. And finally, if you click the submit button and you didn't enter something correctly, we have notifications for you. And so these notifications are all specific to the input that you've entered as a user. And we anticipate that the users, maybe they've entered the wrong strain IDs, or they forgot to check a box that says include copies of my test strains or something like that. And so we are able to anticipate a lot of those errors and make it so that they don't see an ugly stack trace. And so here is just a snippet of the code where we display error messages. And it's really nice because these messages are each widgets of their own, which are by default set to invisible, and so you normally can't see them unless you have some input errors. And so we go through, and if we see any errors, we call display error messages, and then we go through and we set each of those widgets to visible when relevant. And then the other thing I forgot to include is that this tab, we automatically, when they hit that submit button, if there are issues, we bring them to the notifications tab so they can see what they did wrong. So this makes our lives easier, it makes their lives easier. And so this is just an example of what some of those errors look like. So you remember the first tab was the instructions, they read it left to right, and then they automatically go to the notifications tab if something's wrong. So I just went and didn't input a lot of the parameters and got a lot of errors, and hopefully these errors are helpful to our users. So finally, we have the error checking from the input, but we also want to do easy error checking in the output. And for downstream testing, it's really important to have different types of strains to compare your test strains to. So just an overview of some terms, we called the test candidate, like the new strain that you want to test. So you just made some genetic changes to your strain and you want to see how did it do. So we call that our test strain. The parent strain is the strain that test strain came from. So what was the strain before this that you did your changes to? And then finally there's a reference strain, and that can be a lot of different types of strain, but it's basically just another strain that we use to compare to. So we want our test candidate to do better than all of these. So our plate layouts are, we track them in pandas in the underlying code, and this makes it easy to allow the users to do some error checking. So we can just use the apply map function to highlight these wells to different colors. So you can see we just talked about control, parent, reference, wells, and then, or yellow if it's a test candidate. And so we can see what our plates actually look like when we execute these Hitpick plans. So this is an example of what a plate would look like in the output in the Jupyter notebook. And so this, they don't do anything with this downstream. This is only for them to look at now and see, like, does this look like I thought it would look like? You know, did I enter that there would be however many 6 control or empty wells? And did I say that I was going to have 30 test candidates or something like that? And so this isn't the actual Hitpick plan that we give to our liquid handling robot. This is only for their purposes to see, do my plates look like they should? And this is great for building trust in the tool. And so when the users were not so sure about using the tool to begin with, this made them feel more confident about their results. And this is also way better than scrolling through a CSV that says, you know, source well, destination well, source well, destination well. That's not very easy to read. So finally, once we have our Hitpick plan, which is the CSV representation of those plates that you guys just looked at, we are able to send it off to the liquid handling robot that can do the pipetting for us. So this isn't exactly, oh no, will it play? There we go. This isn't exactly Hitpicking per se, but I really wanted to show you guys what it looks like to have a robot do your pipetting for you. So let's see, we'll go to the beginning. So right now it is getting rid of its old tips and putting new tips on, and then it will go over to the plates and do some liquid handling. So for all you biologists that have done manual pipetting, this is really exciting. It can, but that one in particular is not an example of one. I didn't have a good example, so just to sum it all up, Jupyter notebooks have been really invaluable for the solutions engineers. We started off with a plate with samples in it that were similar to each other, and or similar strains were in similar locations, and we wanted to change that. We started off with these scary Excel tools that we didn't really know what to do with, and we weren't really sure how they worked. And we have our scientist who, you know, uses these Excel tools, appreciates the Excel tools, but maybe wants a better option. And finally, we have our liquid handling robot that is there waiting to do our work for us. And that's where Jupyter has become really valuable to us, because we've been able to create a user-friendly interface. We've been able to standardize a solution. So as you saw, there are lots of input and drop-downs so that different teams can use the tool differently, but we're also able to use one tool across different projects. And then finally, we're able to quickly prototype. So we're able to give them this tool, and another great part about it is that we've actually started using this framework for a lot of our other tools, and that's been really nice because, one, we don't have to recreate it, and two, our scientists are kind of used to this format, know how it works, and that makes adoption even better. So I wanted to say thank you, and special thanks to Mark Colangelo, who can't be here today, but he was the primary developer of this tool, and we're really excited about it. And finally, we are hiring at Zymergen a bunch. So if you're interested in a career at Zymergen, you can email hiring@Zymergen. You can come find me later, and we can talk. Thank you. Yes. Yeah, we did. And so there were actually, we used pandas, and there were, depending on the projects, there are different restrictions to how they want them. You know, like some projects will say, I don't want any of the same strains in the same row or the same column. And so, yeah, there was the background code that did a bunch of that arraying. Yeah, so we have an EC2 instance that we run all of these Jupyter notebooks on, and so it's nice to be able to just send them a link instead of telling them to, like, you know, SSH into this machine, do this. And so we simply send them the link, we put it on our Confluence pages, and they just go to it. Yes?
Came down to actually controlling the robot. How was that connection made?
Yeah, so we, the plan that I showed you guys in the beginning that is the, I don't remember what format it is, maybe it's a CSV that has, like, this is my destination plate, this is my destination well, this is my source plate, this is my source well, and this is my volume. We have the, there's automation software that takes in that template and then is able to send it off to the robot to do the work. Yes?
How do you hide all the code when, for the tools that you hand to your scientists? Is it part of the Jupyter extension?
Yeah, so we used, you can make, like, the custom extensions in the, I think you can do it other ways, but we put them in this IPython extension location, and all you have to do is run that magic command that loads the extension and then the name of the extension, and it'll load whatever you told the extension to do. Yeah. Yes?
Have you guys looked at using some of the Jupyter dashboard functionality to do this? It seems like you're kind of in that vein.
Yes, no, we haven't, but luckily I've learned a lot here that I think will help us change some of our notebooks and how they work.
You also go to
Hunt down Pete, okay. Yes?
Is everyone using just like one notebook in like one folder, or is everyone just like copying the same notebook?
Yeah, we make them copy the notebooks for now.
So how do you handle like
What do we do? I mean, we probably just go and get rid of them and have them recopy it, and we probably need to come up with a better solution, but that's what we're doing right now.
How do you, what happens if the algorithm that makes the swap, if it fails, do you show the traceback, or you also have the, like, okay, please contact tech support here option? How do you handle, like, algorithm errors?
Yeah, so those are just a traceback, and we'd have to go back to the code for now and figure it out. Yeah, but that is a good idea. We could put that in pretty easily and just say, contact your friendly developer. So, great. Oh, yes.
What's like the largest kind of
I'm not sure. Nothing too big in Jupyter right now, because most of the solutions engineering projects are the one-off projects. So the data stays pretty small, because normally it's just like, you know, one little project or one scientist that needs something more custom. So, right. Thank you.